Thank you for attending to today's webinar. Today's speaker is Dr. Avi Ma'ayan. Dr. Ma'ayan is a director of the Mount Sinai Center for Bioinformatics and a professor in the Department of Pharmacological Sciences. Dr. Ma'ayan is also principal investigator of the NIH-funded BD2K Links Data Coordination and Integration Center and Mount Sinai Knowledge Management Center for Illuminating the Druggable Genome. The Ma'ayan Laboratory applies computational and mathematical methods to study the complexity of regulatory networks in mammalian cells. If everyone could please mute their microphones to avoid any background noise. And Dr. Ma'ayan, you could go ahead and feel free to get started.
Okay, so I think I'm just going to use the slide that I sent you. So first, thank you for inviting me to speak at this seminar series. Today I'm going to talk about 4 Chrome extensions that we have developed in the past 2 years, and this also enables us to collaborate with the bioCADDIE team. And I guess I can go to the next slide. So the 4 Chrome extensions that I'm going to talk about are called Site Delight, Geo to Enricher, Arches 4, and Datasets to Tools. Next slide. So what are Chrome extensions? So this is the definitions from the Google developer site, and in my own words, Chrome extensions are a small piece of software that can be added to the Chrome browser, and those extensions can enhance the user experience when visiting popular websites. So it can add functionality to those websites by not interfering with the embedded code or not requiring that the host website code is modified. So everything is done at the client using HTML, JavaScript, and other sort of like technologies that manipulate the code on the browser. Next slide. So where can you find Chrome extensions, including those Chrome extensions, some of those Chrome extensions that I'm going to talk about? So those can be found at the Chrome Web Store. On the Web Store, there are extensions, themes, apps, and games. So make sure that you select extensions. Next slide. So the first Chrome extension that I'm going to discuss is called Site Delight. And you just can type in at the Chrome Web Store Site Delight, and then you can find it. Next slide. So this is what happens when you search for Site Delight on the Chrome Web Store. It shows us the first entry. And to install a Chrome extension, it's very easy. You just click on this blue button. And the moment you click on it, it asks you whether you are sure that you want to install it, and then it's installed. So next. So now it's added to Chrome, and you can see that there is a small icon at the corner with the pencil next to this WebEx icon, which is another Chrome extension that I have installed. So now Site Delight is installed on my browser, and I can also rate it. And let's go to the next one. So now I'm going to talk about like what Site Delight does, what functionality it adds. And Site Delight has a relatively few simple features that were added. So for example, if I search for my name on PubMed, I can see the articles that are returned. They have this additional information below each entry from the search, and it says PubMed citation, and then there are 3 buttons, one for RIS, bib, and EndNote. And the moment you click on those buttons, the citation gets exported immediately to your favorite citation management software. Next. So if you click on one of those abstracts that are returned, SiteDelight also has a feature that highlights sentences in abstracts based on their importance. So using a third-party algorithm that it's not our method to decide the importance of the sentences in text, we just use the server to process all of the sentences from the abstract, and the server gives us the importance priority based on the words, based on an algorithm, and then we highlighted the top few most important sentences in the abstract, and this allows you to quickly go through abstracts and find the most important information without needing to read the entire abstract. And you can see also the buttons below. So to export the citation, so the buttons show up both at the abstract pages as well as the search results pages on PubMed. Okay, next. So one of the exciting things about SiteDelight is the ability to cite datasets. And with the BD2K consortium and the links and the interactions that we have, where we try to advance the reuse of data and better extraction of knowledge from data, one of the ideas that are circulating is the ability to cite datasets. So SiteDelight provides the ability to cite datasets. So if you go to a dataset page on the GEO website, which lists datasets for various studies that did gene expression analysis, you can get buttons that also provide citations for datasets. Next. And this functionality is also available on the bioCADDIE DataMed. If you have the SiteDelight Chrome extension installed, then when you search for datasets, for some of those datasets, SiteDelight knows how to process the metadata from each of those entries in searches and provide buttons to export the citations for those datasets. Okay, so this SiteDelight Chrome extension was developed by Esther Chen, who was a summer student, and she is undergraduate student at Cornell at Ithaca. And this summer she's going to do a summer internship at the NIH. And she was helped by Gregory Gunderson, who was a developer in the Ma'ayan lab and now is a PhD student in the computer science department at Princeton. Next. So just if you want to remove some more sort of like background information about Chrome extensions, if you want to remove or disable a Chrome extension that you have installed, you can go to your Chrome extension settings, and then instead of clicking on settings, which what happens when you browse to the settings folder, you click on extensions, and then you can either disable or remove Chrome extension. So for example, now I can remove this SiteDelight Chrome extension if I don't want it to appear on my browser when I'm browsing like PubMed articles. So next. So the next Chrome extension I'm going to discuss is called GeoToEnricher. And again, GeoToEnricher is publicly available on the Chrome Web Store. So you can type GeoToEnricher in the search, and then the only entry that is returned. And then you can click Add to Chrome, and it gets added to Chrome. Next. So GeoToEnricher is also working on the GEO dataset pages, but even after doing a search, so here I did a search for MCF7, trying to find some studies that profile this breast cancer cell type, and you still don't see any new functionality added. You can see the GeoToEnricher icon in gray at the corner, but it's still, you don't see anything. And then you only see stuff once you click on one of those studies. So if you click on if you go to the next slide. So this is a dataset landing page at Geo for gene expression analysis using microarrays. And only when you click on this button called Compare to Sets of Samples, which is a button that already appears on the Geo database for the dataset landing pages, you see this functionality that is provided by GeoToEnricher, which is step 4, extract knowledge with GeoToEnricher. So before you actually click on this new link, this blue link that was added to the page, you first need to select your samples. So if you go to the next slide, if you click on this select which sample to put in group A and group B, you get this interface that is a part of Geo. And here I decided to select, to compare in the first group 3 empty vector control samples for MCF7 cells. And then in the group B, in the treatment condition, I selected 3 samples where the cells were treated with estrogen. And you can go to the next one. So now I clicked on this blue link, and you can see the control samples, the GSM number of the control samples and treatment samples at the top of this table. Then you can have some options of the methods that you use to compute differential expression, and also the cutoff and the normalization method. You can also enter some metadata. So here I put in MCF7 as the cell type or tissue. The perturbation is estrogen, and then or estradiol, and then the relevant disease is breast cancer. And here also I put in a meta tag. So here I created this hashtag or meta tag that is called bioCADDIE_demo. And this meta tag allows you to aggregate many signatures for many studies together, and we'll get back to it in a second. So this is an important feature that allow you to create collections of signatures. So the moment now I have all of the things set up to compute gene expression signature with this Chrome extension, GeoToEnricher. And what it will do when I click on this blue button, it would download the samples and then compute differential expression between the control and the treatment conditions. And then we'll have the differentially expressed genes that are up or down regulated after the treatment of those cells with this hormone or perturbogen, which is estradiol. So let's go to the next. So once I click the button, I had to wait for about 30 seconds, and then a new button appeared, which is this gray button, Open Results tab. And once I click on that, this is the next slide, it takes me to a results page. And this results page lists all the metadata related to my signature. It has link to look at principal component analysis of the samples, also a hierarchical clustering heat map that can visualize the sample, as well as buttons to download the up and down regulated genes, and links to downstream analysis tools, including Enricher, L1000, CDS2, and Paella, which are tools that we've developed for analyzing signatures. So if I click on the up for enricher, this link for enricher that is next to the up, I get enrichment analysis for the upregulated genes. So let's go to the next slide. So this is the result for enrichment analysis, and we clearly see signature for transcription factors that regulate the cell cycle. So those are FOXM1, CHIP6 studies that were previously published and showing enrichment for those genes that are upregulated after estradiol treatment. And if you go to the next one, we see also cell signaling pathways and other pathway databases that support this idea that we have an increase in the expression of cell cycle related genes and mostly mitotic related pathways. So this clearly, and again also for the NCI nature pathways, we get FOXM1. So it's clear that FOXM1 become activated and those cells increase in their proliferation. So let's go to the next. So now we have this tag that I mentioned, and this tag allows us to create reports for collections of signatures with another tool that we've developed called Geneva. So if I go to Geneva, which is the next slide. So this is the website for the Geneva, and I can do a search for all of the collections in the Geneva database. Automatically, this metadata hashtag signature was added to the Geneva database. So in the Geneva database, we have a list of signatures that people extracted with hashtags. And right now, I only have one signature in this collection. And because I only have one signature in this collection, I can't really create a report yet. The report requires at least 4 signatures that have the same hashtag. So let's go next. So this is an example of a report that was created using GeoToEnricher, and it's a report that used the dexamethasone hashtag. And the idea is that dexamethasone is a drug that is highly studied. So there are many labs that use this drug to treat different human cells and then measure gene expression before and after the drug treatment. So what we wanted to do, we wanted to collect signatures from all of those studies and then compare them and create a report for them. So in this particular report, there are 86 signatures for dexamethasone. And in the report, first we have a list of all of them. And each of those links, like this blue title links, take you back to the results page from the GeoToEnricher results page that we've seen before. So let's go to the next slide. So this is part of the report. So in this report, the first heat map shows the genes that are mostly consistently upregulated across all of those studies. So the columns are those 86 signatures that were extracted from using GeoToEnricher. And then the genes, the top 50 genes, are ranked based on their consistency of being upregulated across all of those studies. So that means that FKBP5 is a gene that is almost always, regardless of the tissue, becoming upregulated after dexamethasone treatment. And it's also regardless of the concentration and the time point that the gene expression was measured. So we can also look at enriched terms. So we can look at enriched pathways and also enriched transcription factors that are regulating the up or down regulated genes after the dexamethasone treatment, and this is enrichment analysis using the ENCODE database, and the transcription factors that were profiled with ENCODE are ranked based on their overlap with the up or down-regulated genes that are in each of those dexamethasone signatures. So the top two entries are the glucocorticoid receptors, which is this NR3C1, which is the gene symbol for glucocorticoid receptor, which is great news because dexamethasone is a known agonist of the glucocorticoid receptor. It's an anti-inflammatory steroid that is used in several diseases, including asthma. And this known target is also a transcription factor. And we see that those transcription factor targets, as determined by ChIP-seq, are also enriched for the upregulated genes in many of those studies that were extracted from the GEO database. So let's go to the next. So the final part of the report from Geneva queries the dexamethasone signatures against the L1000 links dataset, where thousands of small molecules were used to treat several human cell lines, and then gene expression was measured with the L1000 technology, and we created a tool that queries those signatures and returns the top drugs that can either reverse or mimic the expression of the submitted signature. And here in blue are drugs that reverse the signature, and in red are drugs that mimic the signature. And you can see that this central cluster has dexamethasone many times as entries. So this is good news because it shows that out of those thousands of small molecule drugs that are in links, many of the studies from GEO match the studies or match the same perturbagen or drug as it was for the L1000 links. So in both experiments or in the same genes go up and down. And we also find other drugs in this cluster that potentially have very similar effect to the dexamethasone treatment effect. And we also see some other experiments that don't show the same genes going up and down. So those can potentially be due to cell type and study differences. They could also point to, I guess, less quality. So all of this idea of reproducibility can be potentially tested by this approach. And we can sort of like rate the consistency of a study with the known biology. We can also identify small molecules that are potential mimickers of dexamethasone. For example, we see some BRD compounds that have no known function, and those could be potential drugs that could be developed as steroid anti-inflammatory alternatives for dexamethasone, which is known to have a lot of side effects. So let's go to the next slide. So both the GEO to enrich a Chrome extension and Geneva were developed by Gregory Gunderson, and there were two papers that describe those two tools. GEO to Enricher was published in Bioinformatics in 2015, and the Geneva system was published in 2016 in last fall. Next. So in Geneva, when we looked at the report, we saw those heat maps. Those are interactive clustering heat maps. And this is a tool that we also developed, and this tool was developed by Nicolas Fernandez, who is a postdoc in the lab. So he's still actively developing this data visualization gadget called Clustergrammer. And you can get the source code and documentation about Clustergrammer and how it can be integrated in your own tools and websites on using those links that we will share. So GeoAutoEnricher and Geneva is only working for microarray data. And the reason it's working on microarray data is because microarray data sets are very organized and already in a relatively straightforward way. You can take the data and you can download it, and then you can process it. And this is much more difficult for RNA-seq. And the reason it's more difficult is because the RNA-seq FASTQ files are very big, and there are no real standards on how to process the data, and the data that is provided has less annotations. But in general, the field is switching to RNA-seq. So here we are looking at the accumulation of the RNA-seq. So on the left are the number of samples that are currently available on Geo and SRA for RNA-seq for the 4 most popular platforms for human and mouse, as well as the number of samples for microarrays from this most popular Affymetrix chip platform. So these are not all of the RNA-seq and microarray datasets, and there are indeed more microarray samples in Geo than RNA-seq right now. But as you can see on the right side, the number of samples that are submitted to Geo and SRA with RNA-seq data is rapidly growing, and there is starting to see a decline in the number of microarray studies that are submitted. Next. So Alex from the lab, he aligned all of the align and process all of the human and mouse RNA-seq samples that are currently available on Geo and SRA. And this is just some neat analysis that he did to look at the quality control. And this is looking at how many reads that were present in the FASTQ file were able to be aligned to genes. So this tells you the quality of the alignment, and this is the quality by country, as well as quality by zip code. He also did quality by institution, but that could be a little bit sensitive to show. But it's an interesting thing to see that there is high variability with the quality, and the difference in quality can also be due to the type of samples that are being processed. So if you use, let's say, post-mortem tissue samples from humans, you are likely to have RNA degradation and less quality regardless of the quality of the sequencing service, but it also could be some issues with the cores, the sequencing cores at the different places and their methods of practice, and that could give them some feedback on where they stand in regards to quality. Next. So through this Chrome extension, the third Chrome extension that I'm describing today, Arches 4, we provide access to all of the process data that Alex generated through aligning all of those RNA-seq datasets. And this Arches 4 is available on the Chrome store. It was recently added, and it looks like it has 32 people installed it, so it's hasn't gained that much popularity yet. And let's go next. So here, for example, if I'm looking for studies that apply dexamethasone and then measure gene expression with RNA-seq, I get 229 studies. And then once I click on this and I go to one of those studies, and you can go to next, the Chrome extension adds two things. It adds a link, like if you can see it at the corner, it says now with counts, and it provides a link to a serious matrix file. So this is a text file that has the gene as the rows, and then the metadata of the samples and the GSM IDs as the column. And then it also adds this clustergrammer visualization of the data that you can potentially start interacting with. And here, for example, this is a study that used TGF-beta and dexamethasone, and you can see that the samples are clustered into two groups. The first 4 samples are mock-treated cells, and then the rest of the samples have relatively similar expression profiles, but you do see that the last 3 samples are slightly different. And you can start looking at and exploring the data using a clustergrammer, which is taking the data that we processed and just provide access to it immediately. And using clustergrammer, you can zoom into clusters. So you can use this slider that is shown on the top right, and then find a cluster. And once you click on the trapezoid next to the cluster, you can get the list of genes that are associated with the cluster, and you can pipe those genes to enricher. So if I go to the next slide, this is using different categories of enricher and looking at those genes that are belonging to the cluster that I showed from the previous slide. And we see that there are a lot of genes that have extracellular matrix components and fibronectin binding, as well as TGF-beta studies for microarray experiments that we extracted from GEO. So there is some consistency and confirmation that we get some of the same differentially expressed genes, or even though we haven't really performed the differential expression analysis yet. So next slide. So Alex is in the process of building a website that will provide access to all of this data, and you'll be able to analyze the data and get a slice of the data. So on this site, you can actually put any type of metadata search term and obtain a subset of samples that have the meta tag associated with those samples. And then you can download the samples and potentially use that to identify average expression for genes. You can impute knowledge for genes, as well as do other things with this data that we have some ideas of how to make this a usable resource. Next. So Arches 4 is actively developed by Alexander Lachman, and the Chrome extension was developed together with Dennis Torre. So next I'm going to talk about the last Chrome extension, which is Datasets to Tools. And the idea behind Datasets to Tools is that there is currently no systematic effort to associate datasets with tools, and there is a lot of our tools that have preloaded data, So the tool has a unique URL it's a permanent URL. It has the data already loaded into the tool, but there's no real way to search for it and access all of those analyses for reusability. In general, in the field, there are databases that list tools and databases that list datasets, but there is not really a way to associate tools with datasets. And if this can be done systematically, we potentially can help with knowledge extraction and accelerate discovery. Go to next. So we came up with this concept called canned analysis. So this is a potentially citable digital object that link a dataset to a tool and provide some potentially interactive web-based analysis of the dataset. Next. So each canned analysis record in this database of canned analysis will have a link to the dataset, the original dataset that has been analyzed already, the tool that was used to operate on the dataset, and then the canned analysis URL, and some annotations, so description of the canned analysis. And currently, this is working on the Geo database as well as DataMed. So going back about talking a little bit more basic stuff about Chrome extensions, you don't have to have the Chrome extension available on the Chrome store in order to install it. So there is a way for developers to upload and install a Chrome extension using the settings in Chrome. So again, you can go to this extension tab under settings, and then you want to make sure that you check the developer mode, and then you can browse to when you click on this load unpacked extension, you can browse to a folder, and here I'm browsing to the folder where Dennis provide me with this Chrome extension, the Datasets 2 Chrome extension, and I select the folder, and then I click OK, and voila, the next slide shows that the extension is installed and enabled. So we will provide the people on the call with this extension. I'm going to provide a URL. It's in the slides also, that people can start playing with this Chrome extension. And once it's installed, if you go to the next slide, and you go to a database like the Geo database, and you do a search, like for example here, I'm searching for MCF7, I get the same study that I analyzed earlier, and all of a sudden I see below the link to PubMed and PubMed Central, I get this box that gives me this Datasets 2 tool, new functionality, and it has links to some analysis tools. So next slide. So here I clicked on the L1000 CDS square green icon that was in the middle, and it gives me a list of canned analysis that were performed with this tool L1000 CDS square, which is this tool that query signatures against the L1000 database for finding small molecules that can either reverse or mimic expression. And I have some buttons here. I can get directly to the canned analysis by this link column, and I can also share and I can read more information about the canned analysis. I can share the URL of the canned analysis, and I can embed the canned analysis in my web page. So if I go to the next slide, here is an attempt to find estrogen mimickers. So those drugs that are ranked by the L1000 CDS Square app are prioritizing small molecules that have a similar effect on cells, and the top two entries are MCF7 cells, that basically those drugs turn on and turn off the same set of genes that estradiol is turning on and off, and those are potential estradiol mimickers. And the broad compound, for example, is not a well-studied compound. Next. So the same box of canned analysis shows up on DataMed when I have the Chrome extension installed, and I'm going to the DataMed search engine, and I'm looking for MCF7, and I'm filtering by studies from GEO. And the idea is that we will have more canned analysis added to the database for other databases, not just the GEO. Next. So this Datasets to Tool is actively being developed by Dennis Torre, and Dennis created a website that he demoed to the bioCADDIE team recently, where he natively embedded the canned analysis in the bioCADDIE database, so you don't have to install the expression. So if there is a decision that we like this additional feature, it's very simple to simply add this to the native code base of DataMed, and this really enables us to work together on this project without a need to share the code and overlap our efforts. So we could work independently developing this extension, and then the transition to a native code should be very straightforward. The URL below can also give you the access to the Chrome extension. So this is the next slide, just a summary. I showed 4 Chrome extensions that we've developed. This is sort of like a new way to add functionality to existing sites, and I think it's a relatively cool new direction for software development. A lot of people are using the Chrome extension, the Chrome browser, and it enables us to potentially add functionality to popular websites where people access the data, and the key is capture the processing of the data sets by users and then reusing existing analysis. So since I have a few more minutes left, I'm going to, in the next few slides, I'm just going to show you how to get started with developing Chrome extensions. I'm not a very, I guess, polished developer, so I'm going to pretend to be one. But to just get started, there is a site, or a very user-friendly site at the Chrome developer site, where you can read about how to build Chrome extensions. And if you are following this demo, they provide under resources 4 files that you can download, and you just need to download those files, put them in a folder, and then if you go to the next slide, so here I downloaded those 4 files. I put them in a folder called demo extension, and I edited the popup.html to just be a very simple web page that says hello world. So I took out all of the other stuff that was in there and just created a small web page. And then the other important file is this manifest.json file, and it just has some metadata about the Chrome extension. It has information about the name of the app and what it does, the version, where is the icon, and what it does. So this is obviously can get extended, and so once I have those 4 files in this, I don't really need the popup.js file because I took the code that calls it out from popup.html. you go to next, I browsed the same way I installed Datasets to Tool earlier to the folder that had the demo extension, and I just selected the folder, and then it added this extension, Getting Started Example, and then it added the icon hello in the right corner, and then if you click on this icon, you see hello world, which is what's in the pop-up.html file. So I would like to also give special thanks to Kathleen Jagodnik, who helped us a lot with many aspects of all of those 4 projects. And Kathleen joined us this week as a postdoc, and also the other members of the lab. And here you see a lot of the summer students that joined us last summer, and Esther, who developed SiteDelight, she's in the center, standing in front of me. So that's it. I think we have a few minutes for questions.
Are there any
Avi?
questions for Dr. Ma'ayan?
Yes.
So, Avi, one of the questions I would have, this is incredible. I mean, this is just a wonderful suite and a great set of data, and greatly appreciate your introduction. Now, the question is if as people start to use this, are you willing to have people come and spend a brief time with some of the people in your lab working through the challenges?
Yes, absolutely. So we would like to host people here. We have some space, and that's yes. If you have someone that is interested, send us an email, and we'll have them here. And there's some housing possibilities also. So yeah, we are willing to accommodate such requests, and I'm sure it'll be like beneficial for both sides.
Yeah, combining this with your presentation at the Links meeting and the way this stuff is coming together, the AACR meeting, this is really an incredible resource and a lot of integration that's hard to do otherwise. Thank you.
Thank you, Gordon.
Are there any more questions? Okay. Well, thank you so much, Dr. Avi Ma'ayan. Please, everyone, so the next webinar will be on May 11th at the same time. And thank you for presenting this time.
Oh, you're welcome. It was great. Thanks for having me.